Hello, everyone, and welcome back for part 2 of our webinar series, Cryo-EM Back to Basics. So for today's session, we will be covering Cryo-EM data processing and interpretation. If you're able to join us next month, we'll be focusing on some of the common limitations in Cryo-EM and how to overcome them. So before we begin, I have a couple quick housekeeping notes. So today's webinar will run for 30 minutes. And after, there will be a 15-minute Q&A session. So just like last time, you can submit your questions in the Q&A box in your toolbar. If there are any questions that we do not get to during this session, we will follow up with you next week. Also, as a reminder, a recording of this webinar will be sent to anyone who registered. So now it's my pleasure to introduce our speaker, the CSO of Nanoimaging Services, Giovanna Scapin. So Giovanna has extensive industrial experience in the field of structural biology and structure-based drug design from her career at Merck. She trained at the New York Structural Biology Center in order to learn cryo-EM, and then played a pivotal role in establishing Merck's cryo-EM facility. Since joining NIS in 2020, she has been instrumental in advancing cutting-edge research in this field. So without further ado, please join me in welcoming Giovanna Scapin. So Giovanna, over to you.
Hello, everybody, and welcome to part 2 of this 3-part seminar in which we will discuss data processing and interpretation. As I mentioned at the beginning of the first webinar, the intent of the series is to provide you with some of the very basic knowledge of the theory and practice of cryo-EM. And for a more detailed and in-depth description and discussion, and all the theory that's behind all of it, I will urge you to refer to the many online tutorials that are available and to the wealth of articles that are available. The manuals for the software that are used for data processing are also a great source of information. This is a brief overview of what I will cover and discuss today. And I will start with what's in an image, which we have seen in the last webinar. We'll talk a little bit about the different stages of data processing, from pre-processing all the way down to 3D reconstruction. And at the end, I will briefly discuss the conformational variability analysis, how we look at it, and what are the information that we can obtain from that analysis. So let's start with a brief recap of what we learned last time, from sample to micrograph. So the sample is in solution, is tumbling around. We put it on a grid, and then we flash freeze it into a thin layer of ice. This frozen grid is moved into a microscope where we take many thousands of images of our sample. Now, you probably remember these images are 2-dimensional projection of the 3-dimensional object that we have in our solution. So what the micrograph at the end of the day shows are these different 2-dimensional views of our object. And this is the data that now we have at hand, and this is the data that we need to process and interpret in order to get to the final result. Some useful terminology when we are thinking about data processing, or even when we are just looking at the micrograph during data collection, there are a couple of terms or definitions that might be useful to know. One is power spectrum, which is basically the Fourier transform of the image. And by looking at the power spectrum of an image, we can have a good idea of how good or how bad this image itself is, even if we can't see too many details in the micrograph themselves. So we have a really not so good image, a good image, and basically the more ring we see in the power spectrum, the more information that the micrograph contains. And the more micrograph like this we have in our data set, the better the data set, and the more details we will be able to see at the end from our sample. There are other information that the power spectrum can provide us, and for example, can inform us of problems with the microscope, such as stigmatism, for example, or problems with the grid itself, the crystalline ice, as we can see on the bottom right. These may not be immediately visible from the micrograph, but they are clearly visible if you look at the power spectra. And monitoring these images can really help us identify issues early on and likely address those issues before we can waste an entire day during a data collection that we won't be able to use. Another term that you all have heard of is what's called CTF, or contrast transfer function. And it derives from the fact that all the images collected by a microscope are basically corrupted. And this corruption is actually a fundamental aspect of the transmission electron microscopy. And it causes an uneven transfer of information as function of a spatial frequency. This transfer can be mathematically described as a wave. And in standard transmission electron microscopy, the CTF starts at 0 at low spatial frequencies and oscillates between positive and negative maxima throughout the spatial frequency. Zero crossings, like this, would intersect the zero line, are areas in which we have absolutely no information. The CTF is also modulated by what we call envelope functions that derive from a spatial and temporal incoherence of the electron beam. And the result is that they dampen the CTF at high spatial frequencies. Oscillation between the CTF are easily seen, guess what, visualizing the power spectrum of the cryo-EM images. These concentric rings that we call tone rings represent alternating high and low contrast transfer as a function of spatial frequencies. Now that we know what power spectrum is, these are essential tools that will help us in addressing the beginning of the data processing workflow that we call pre-processing. Pre-processing includes all the work that is done to evaluate the quality of the individual micrograph, to select the good micrograph and exclude the bad micrograph from further processing, and to prepare this micrograph for the entire processing workflow. I also include the very initial particle picking in the pre-processing set of the analysis. In the large majority of software that we use nowadays, pre-processing is fully automated, so there is no human intervention required, and it can be executed on the fly while the data is collected, which saves hours, if not days, on the entire process. The first step of the pre-processing is the frame alignment, and we know why we have to do it, and we know where is this coming from. We already seen it, that in order to get high resolution information, we need to work with the high exposure and long exposure times. But these two factors induce a physical drift and a beam-induced motion of the particles. And as we have learned in the previous webinar, if you take a still picture of a moving object, we obtain a very blurry image with loss of resolution. But the introduction of high-speed cameras a few years ago allow us for a different type of recording, not a still image, but a film, a movie, in which up to 50 frames can be collected in a short period of time. And these frames can then be internally aligned, resulting in a much clearer and more detailed image. So this is step number one. Step number two is take these aligned images and estimate the CTF parameters. And the goal here is to enable a computational correction of the CTF for each micrograph. The CTF, I didn't mention it before, is a function of many, many different terms, and including the electron wavelength, the spherical aberration of the microscope, and the defocus that we set the image collection. Within a single data set, the electron wavelength and the spherical aberration are constant, but the defocus will vary. And so the most important task of the CTF estimation is basically determining the true defocus of each image. In order to do so, the tone rings are radially averaged, and we generate a plot of the CTF as function of the spatial frequency. There are the CTF estimation programs that then will fit the observed CTF, and so the tone rings, to the theoretical model, the green lines over here. And the quality of the fit is then assessed by cross-correlation. CTF modeling is also a very effective way to access the micrograph quality in a very objective manner. The fitting reports the resolution in which the data no longer fits the model, which basically corresponds to the point in which no more information can be extracted from the image. The cross-variable correlation and the resolution limit of the CTF fitting are very useful metrics to exclude poor micrographs from the whole data set, as can be visualized, for example, using CryoSpark Live on a graph similar to this. The last part of the pre-processing is, as I mentioned, particle picking. We remember now that we collect 2D projection of three-dimensional objects, but how do we go from this 2D projection back to the 3D object? I will use something different from a protein to explain the process, and not the protein that you can see here, but a duck. And this is a very clear tool that I found very, very useful when I was looking at this article, which is highlighted in the bottom, and that I read when I was trying to understand how this works. So the duck is our object, and a micrograph contains multiple views of the duck and other spurious artifacts that are due from ice contamination to debris to anything else. The first step is to identify all of these objects on the micrograph and extract them into a particle stack, which is basically just a list of particles with their coordinates, X and Y, and the micrograph that is associated to each particle. This process is called particle picking, and it requires a little bit, we should look at it a little bit more. One big thing is that because of the noise, the low signal to noise level and the poor contrast, particles are often very difficult to see in an image. And the first thing that the software do in order to make them more visible is to enhance the signal by applying a low or medium pass filter, or sometimes a denoising process. At this point, the particles are a little bit more visible, and we can start the picking process. There are three ways in which we can pick particles, select the particles. The first is manual picking, which basically nobody does anymore. It's because the process has been completely and fully automated. If you want to, you can still use manual picking, maybe to select a few hundreds of particles that then you can use to generate templates to proceed with a much more automated process, but really, why would you ever do it? What we do, if it is the first time that we see a sample of which we don't know the size, we don't know the shape, we don't know anything, we use what is called a blob picking. The software picks out all the blobs that are of a given size range, so between 50 and 150 angstrom, for example. This is a very unbiased process because it will pick anything. But since it will pick anything, there will probably a lot of contaminants and spurious particles that are not really our sample, but they are just junk particles that end up in our data set. Very often, blob picking is used for the first 100, 200, 500 micrographs. And then from these picks, we will calculate a set of 2D classes. And these classes are then used for running what we call a template picking. The size, the classes now contain information, size, shape, orientation of the objects that we are looking at. And they can use by the software to identify other particles in the micrographs. Usually there is a correlation score that is set to match the templates to the particles that are picked. And this is to maximize the number of good particles and to minimize the number of junk particles that are selected during the process. Template picking has a problem, which since we are driving the picking with something we know already of, it can introduce some bias in the final particle stack. And this can be a problem if, for example, the sample that you have has some orientation issues, because you are kind of exacerbating the particle that you are picking using a template. In the past couple of years, a lot of, no more than that, 5 years, a lot of machine learning approaches has also been developed. In our experience, a well-trained machine learning-based selector system has the highest accuracy for most data sets. So it's definitely worth to do maybe one first round of picking using blob or templates, generate a particle stack, and use it to train the machine learning approach, and then rerun the picking with the machine learning topaz cryo or anything like that. Now we have a particle stack, we are ready to move into the real processing, which is what I consider real processing, so from the initial 2D classification all the way to the final 3D map. For the 2D classification, as I mentioned, we start with our original particle stack, and the first step, as the name says, is the classification of all the particles in groups of similar particles. So, for example, here we put all the size particles, all the size duct, side view of the duct together, or the front view together, or the top view together. And of course, we generate stacks of particles that are just junk particles, and we will remove them for further calculation. The second step involves averaging all these particles after orienting them in generating the final 2D classes. This process, the averaging, of course, enhances a lot the signal to noise and makes details visible. So, for example, now we can see that the duct has eyes and a beak, but it's still an averaging process. And so if the particles that belong to a given class do not align very well because they are poorly defined, because they are too small, because they are flexible, or because they are otherwise different from each other, the resulting image will not be that sharp, and it will be still a very confused and not very clear image. I have some examples of the 2D classes, and I want to use them also to show what we can learn from the classes. I wish that all my sample would look like this. It's very sharp images, secondary structure details are visible, multiple views. This is a very good sample, and there is probably a three-dimensional map in a not so distant future. On the other hand, these particles are really small, they're really fuzzy, and the sample can be either too small or maybe it's falling apart. This is telling me that we cannot proceed for high-resolution structure determination, that there is a lot of work that needs to be done. This sample is also very fuzzy, which implies flexibility, but the particles are also of many different sizes, which suggests that there is a lot of heterogeneity. And again, it's not a good sample to move on for further 3D structure determination. This last set of 2D classes are as good as the first one. They are very sharp, they have a lot of details, but there is only one, maybe two, views of the system. It means that a sample suffers of what we call preferred orientation, and that we need to solve this problem before proceeding to high-resolution structure determination. In any case, there is plenty of information that can be derived from 2D classification. And in many cases, the data processing stops here, stops at the 2D classes, because maybe we are looking for information that are not the three-dimensional structure. It might be something on which detergent do we want to use to minimize or to resolve the preferred orientation issue, or which grid type is more suitable to maintain the integrity of our sample, or which data collection strategy we want to use. The 2D classes can really guide us on what to do next. And this next may be going back to a more stable sample or a better grid, or hopefully to proceed with what we really want, which is the 3D map reconstruction. So let's go back to our doc to understand the process of 3D reconstruction. The Fourier transform of the 2D class average is a slice of the Fourier transform of the initial object. So if we combine this section in reciprocal space, we obtain the Fourier transform of the original object. So the inverse Fourier transform of the combined section generates then the original object. And of course, the more views we have, the more information we have about the three-dimensionality of the object itself, and the better our reconstruction will be. So we need the 2D classes in order to generate the individual Fourier transform that then we recombine to generate the final object. There is something that we need to remember. This is not a perfect world, and mistakes are made and errors are intrinsic in our workflow. For example, particles that kind of look similar, but they're not entirely similar, but they're still somewhat similar, they may be pulled together into the same class. And so the same class will have some errors that then get transferred into the reciprocal space. The other very important thing is that we will never have the total, complete, infinite number of possible 2D projection. We are always going to have a subset of this possible 2D projection. And so now there is an interesting fact, because one three-dimensional object can give you one set of 2D projections, 50 2D projection or 10, but there is a one-to-one relationship. But the same incomplete set of 2D views is, when back projected, can be consistent with many different three-dimensional objects. This is called ab initio calculation, and it's why during the process of back projection, during this process, we reconstruct many, many different 3D objects, and we analyze them, and we evaluate, and we compare. And at the end, there will be one that is the most likely to represent our initial object. Since we never have all the views, the initial map generation is a trial and error approach. And it should be recomputed many, many times, changing parameters, changing the number of classes that we're looking for, changing the resolution limit, the number of cycles, until we get to a consensus model that can represent a reliable starting point for the 3D reconstruction. The 3D reconstruction can be as simple as a couple of refinement jobs, and I'm using here the terminology that we have in CryoSpark because that's what we use to do all our analysis and work, and a final post-processing set of polishing jobs. Or, depending on how it goes at the beginning, it can be extremely complicated and require many, many different steps. For example, we can be not very happy with the results of the initial homogeneous refinement, and we decide that we want to go back and do some more clear up of the particles in 2D before generating another ab initio map and going around and around in this circle. Or maybe we are pretty happy with the two particles that went into this class, and then we're going to try to sort them into three-dimensional and use a heterogeneous refinement or 3D refinement, 3D classification in order to further sort the particles. And maybe we can even put some conformational analysis in at this step. We can add some very complicated or more sophisticated computational. And sometimes we step all the way back to the picking and extraction step. We can change the box we use to extract the particles, or we can use different particle tools, particle picking tools, to see if we can get a better set that will give us a good result at the end. The bottom line is that there is no definite pathway to go from a 2D class set to a three-dimensional map. And I'm not even sure we ever get to a point that can be called the end. If you ask anybody that has been doing the processing, there is always something else you can try, something else that you can do. But eventually we need to stop. And it's the result, it's the map itself that guides us to this final call. But we need to be able to understand what the map is telling us, basically. So let's talk about resolution, for example, because everybody talks about resolution. And we put a lot of emphasis on the resolution values. But do we know what they really mean? In X-rays, the resolution D, it's the mathematical definition and corresponds to the farthest point spot from the center we can see on a diffraction image. So, for example, this crystal diffracts to better than 2 angstrom resolution. But in Cryo-EM, the resolution is a correlation between two half maps. And this correlation could as well change depending on how the half maps are calculated. So to me, resolution in Cryo-EM is never an absolute number. And I don't try to get hung up on it. And most importantly, I try not to go just by that number. I will always look at the map and what the map is telling me. There are another couple of things that I think are important. The first one is that EM, electron microscopy, is a direct imaging technique. We have the intensities and we have the phases. And so there is no phase problem like in X-rays. And so there is no error introduced by the phase problem. And what happens is cryo-EM maps are usually better than X-ray maps at comparable resolution. In addition, what is normally cited is the overall resolution. But what is really important is the local resolution, which can vary a lot depending on the sample or the stability. So for a map that, like this one, had an overall resolution of 3.5 angstroms, there are actually areas which are below 3 angstroms and areas which are well above 4 angstroms. Since the resolution is basically an estimation of how well two half maps agree, resolution areas indicate spots in the 3D reconstruction where the agreement is poor, which in general can attribute to the presence of, for example, conformational flexibility or disorder. This is already a very important information that we can get from an electron microscopy reconstruction, even if the resolution is not higher. Very high cannot allow for atomic details, we can already have a feeling for how this molecule moves, for example, or what are parts that are more or less all there. So conformational flexibility is something I want to spend a little bit of time in the last couple of slides, because I think it's very important for a variety of reasons. This is the greater complex, the structure of which was published in 2024. I'm going to use this as an example because I think it's easier to understand what I want to say if we look at things. So this map was a nominal 2.9 Angstrom map. That clearly is not a 2.9 Angstrom map. And also there is a lot of variation within the map itself. It looks okay at the beginning, but it does have a lot of disorder at either end. So because of the quality of the map, and because we know that the graders tend to be very complexes, tend to be very, very flexible, we did run a 3D variability analysis, always using CryoSpark. And this resulted in the identification of several subclasses in which the relative position of the 4 or 5 domains in which you can split the original complex is very, very different. Now, there are a lot of things that you can do with this model. You can put this map together and generate a model that clearly highlights the flexibility of the system. This has a lot of value at the nanometer scale when we can see motions that can be relevant for biological and biochemical reactions. And we wouldn't be able to see this motion in any other way but by collecting a cryo-EM data set and doing a variability analysis. But if you are interested in atomic details at Angstrom level, then we need to focus on one of these classes and try to get the most out of it, doing with the full reconstructions. Chances are that the resolution, the overall resolution will be lower because there are far fewer particles in the final set than there were in the initial set. But it will be, the map will be much cleaner because there is a much better consensus among the particles that went into the map. A local refinement of the portion of interest, which is usually done by masking everything else out but the portion that we are interested in, and focusing the 3D alignment and refinement only on that portion, can usually bring up the atomic details that we are looking for. In my mind, 3D variability analysis is a fancy 3D classification. We group the particles in different classes, not to eliminate them, but to generate many views of the initial system, to generate many classes that have much more information than the single class that we started with. Technically, it's possible to sort the particles in a way that will allow to capture every single conformation present in solution, a true single particle analysis. Practically, this is not possible. Once, for example, because we need a certain level of details to differentiate the different classes. And then because if you want to proceed with a full 3D reconstruction, we do need a certain number of particles within the classes. But I think it's something the field should move towards. And the availability of all these tools is becoming more and more important. And because we all know that nature is not a snapshot, right? But it's in constant motion. And the possibility of analyzing diverse modes and different structures provides us with a much better understanding of all the processes. As I always say, all micrographs, everything is in there. All we have to do is find to extract all the information and visualize them to us. And with this, I will end this webinar and I will take any questions. But before that, let me remind you that the last webinar of the series will be on March 25th. And we will talk about limits and opportunities in cryo-EM. And thank you for your attention.
All right. Thank you so much for a wonderful talk, Giovanna. I really appreciate it. I think we all appreciate it. So we will now go into the Q&A portion of this webinar. So just a reminder, in your toolbar, you will see an area where you can post your questions, and we'll be answering those questions live. So our first question is from Mahesh, who asked, given the Einstein from noise stories, how risky is it to generate down-filtered templates based on AlphaFold predictions?
It's risky, but not that risky. The 2D picking is done at resolution levels that are much lower than what we think about atomic resolution. And so you're ended up working with an envelope, right? You don't really see where a domain is specifically or where a piece of a secondary structure goes or anything else. It does have the risk because you are kind of biasing the set toward a certain configuration, but I think that it's a limited risk. I tend to not do it. I tend to just let the 2D picking work on its own. So I would just pick a lot and do an initial classification and generate templates for that, and then do another classification, and then maybe go into some machine learning or something similar based approaches, rather than generate templates from something that I think it should look like my structure, but I'm not sure. But if it's not there, it's not there, and you won't be able to figure it out what it is. Again, I don't think it's too risky. I wouldn't be my first choice.
Our next question is, there are some publications that report structures where they skip 2D classification and proceed directly to 3D. So what might be the advantages of such a workflow?
So classification in 3D is actually more accurate because you have many more parameters that you evaluate in order to group particles in the same class. And so information that are three-dimensional way are lost on a 2D classification, but are captured in 3D classification. Skipping 2D altogether can be a little time-consuming, I would say, at the beginning at least, because when you do the way we do things, that we run in automatic 2D picking during the data collection, we usually use very loose parameters so that we are almost sure that we pick anything that has a signal on the micrograph. And so we ended up with very large data sets of 2D particles, and doing a preliminary 3D classification, 2D classification really helps out in cleaning up and discard all the junk particles that you have in there. Then you can move directly into 3D. It's actually something that I have done in specific cases. For example, if I know, or if I notice that doing the standard process, my map at the end ends up being preferred. Not terribly preferred, but clearly there are some views that are missing when I do the reconstruction following the standard cleanup in 2D and then 3D reconstruction. So what I do, I will do the first cleanup in 2D just to get rid of the junk, and then take my models and do a series of hetero refinement or 3D classification. And that really works well in extracting from the data particles that are of different orientation, even the one that in subset of very small amount, you end up picking them up. So it could be really useful in case you're looking for very rare views or things that could get lost into the 2D classes.
Our next question is, in a structure-based drug design project where the binding site is visible but there's strong preferred orientation, would it be possible to proceed, or is the data more or less not useful?
Depends on the information that you want to get out of that. So if you see the binding site and you see a series of blobs and you kind of know that things are there and you can put there, but how confident are you that that is exactly how the residues are oriented, how your compound is oriented, the shape of the compound and everything else? So if all you're interested in saying, okay, I know where the binding site is, I know something is bound, that's okay. But if you really want to, and if you're talking about structure-based drug design, you really want to get atomic information about the interaction that the compound makes with specific residues, that the real orientation, the side chains and everything else, then a map that's really seriously preferred is not enough. It won't have enough information for you to be able to make those assessments in a confident way. So sometimes it's much easier to just spend a day on the microscope trying to optimize the preferred orientation experimentally during a detergent screening, during anything, or during just a tilted data collection, than spending weeks trying to beat your model building to a point where you can actually be confident that it looks like something that you want.
So at the end of your presentation, you touched on conformational variability. So Ben asked, given conformational variability in 2D classification, how do you know if a class is a new view or a new conformation? And are there any algorithms for determining this?
In 2D classification, it's really hard to make any judgment, I think. You just take the 2D classes the way they are, and then you move into a three-dimensional space. And that will help you a lot in understanding if it's a different view or if it is a different conformation altogether of the particles. If the conformation is very different, then yes, then you will be able to see, even in 2D classes, that you have different, I don't know, complex formation, for example, or different configuration of your SEM sample. For example, in the slide that I presented, where I had that particle that had 654 different blobs, different components, you can see that. That's definitely not different views. They are different particles. They are conformationally different particles. But if the conformational difference is more subtle, like in the case of the degrader, you cannot really distinguish that at 2D level. There is not enough resolution, meaning there are not enough details in the 2D classes to allow to define the different components. You will always have to go into 3D.
So next, April asks, what is a mask and how is it used in local refinement?
Yes, good question. Because I had no idea what a mask was when I started working in Cryo-EM. So a mask is basically an envelope that you draw around the portion of interest that excludes everything else. So if you're thinking about that, the degrader that we were seeing before, for example, your portion of interest is maybe the interaction between two domains. And so you draw an envelope around that area and you tell the program that it's doing the alignment, align all the objects that are within that envelope and ignore everything else. If the program tends to maybe make an average alignment of all the things because they are in a different position, if you use a mask, it will try to align things within that mask and then let everything else move around a lot. And so you have a much better alignment in 3D and then a much better resolution, but only within that specific envelope that you are defining. It can be too small because you do need to have a certain amount of volume or mass, if you want, in order to be able to align. So technically, you could align a loop, build an envelope around a loop and say, I really want to see what the conformation of this loop is. In reality, you can't because there is not enough mass in the loop per se, if it is a 10 residue polypeptide, to drive the alignment at that point.
So the next question is about kind of particle shapes and the best way to handle processing. So what is the best way to process a data set that contains particles that have a large diameter in one orientation and a small diameter in another orientation?
So I'm sure that there are specific ways in which you can design boxes, especially for particle picking, that are not round or not square, but they are elongated or oval or something like that. I've never had cases that were that extreme. When I had different things that were longer in one direction and shorter in the other, what I always try to do is to get my box big enough to cover the entire the longest length of your particle and make sure that I capture the entire dimension on that direction. That can be problematic because if the particle is really elongated, you end up with huge boxes that can cause severe problems from a computational point of view. It can slow down the processing and a lot of other things. I can look and see if I can find specific tools. Again, I haven't used them, so I'm not really aware, but I'm more than happy to follow up on it and find something that can be useful.
And I'm sure we have people here kind of at all levels. Do you mind touching on what is a box size?
Oh, a box size. Yes. When you extract the particles and you build those 2D particle stacks, you extract the particle, but you don't extract, meaning you isolate the particle, but not just the particle per se. You isolate the particle in a certain amount of background surrounding the particle. And that is the box. So it's the square that is drawn around the particle and as much background as you want. There are pros, pluses and minuses on the box size. There are some empirical values to evaluate the box size, and it used to be 1.5 the size of the particle in the biggest dimension. I'm sure they change. It depends on how the data is collected. It also depends on how big or how small your particle is and the amount of computational effort that you want to put into there.
The next question might be a bit of an opinion question. So Mahesh asks, what is the state of the art for processing software? And are there any advantages with Relion over CryoSpark and vice versa?
Okay, it's a $1,000 question. I don't know. CryoSpark is always been much faster than anything else and has been really designed to do everything, or at least all the pre-processing portion, in a completely user-independent way. We are a CryoSpark shop. We only use CryoSpark. We do have Relion. We don't use it for pre-processing. It's a little bit more complicated to set it up. It's not as user-friendly as CryoSpark is. I'm sure the results at the end of the day end up being the same. But if I have to, in the consideration also how much time it's going to take me to set it up, how easy it is, how many things I have to remember, how many notes I have to put on my computer, I would definitely always go with CryoSpark. Downstream from the pre-processing, they can be, can be equal or different depending on the complexity of your problem. And each software has its pros and its cons on both sides.
Sure. We are getting towards the end of our time, so I'll ask one more question. So there's a question, will binning affect 2D classification for particles that have less than 100 angstrom diameter? And then maybe you could also touch on just looking at small particles in general. What are some of the challenges with data processing?
My experience is that binning doesn't do anything unless you move onto 3D. And then it really affects the final resolution of your system. But in 2D, it's fine. It's not going to affect anything. You can do all the binning, even if the particles are small. Although when the particles are small, you end up using a much smaller box size. So binning can speed up the processing, but sometimes it's just a personal choice if you want to use it or not. The other thing was, how do you deal with small particle, correct, Sarah?
Yeah, or I guess where did the challenges arise with data processing?
Everywhere. Small particles are difficult to see to start with, and so particle picking can be a little bit complicated. You may end up collecting a lot of junk, thinking that they're actually particles, and then that requires a lot of cleanup in order to get a decent, reliable data set. If they're really small, you can classify them accurately in 2D, because they all look the same. It's really hard to decide if a particle is like this or like this, or maybe a different view. And so you end up with classes that are mixed, probably. So particles that kind of look the same but aren't exactly the same. In that case, the 3D classification works much better. So I did have a sample in which it was a very tiny particle, and I did a template picking. I end up with many, 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 many particles. I did a first cleanup in 2D, and then what got me to the final structure was doing multiple rounds of 3D to clean up the particle set, pick the right one, and everything else. So I would definitely focus on a 3D, not reconstruction, but classification more than a 2D classification when the sample is very small. And also I will use a relatively large box size, larger than what you think could be the required number or something like that, because that also helps a lot with getting the resolution details set.
Thank you. Well, with that, we are at the end of our time, and we will conclude our second webinar from our series Cryo-EM Back to Basics. So I would like to thank Giovanna again for a wonderful talk. I want to thank all of our attendees for joining us and for your thoughtful questions. So there were some questions that we didn't get to, and for those, we will follow up with you via email. And just a reminder, anyone who registered for this webinar will have access to a recording. So our last session for this series will be on March 25th. So that's a Tuesday, just a reminder because it was changed. And so during that session, we'll be covering topics on common limitations in cryo-EM and how to overcome them. If you have any questions or you'd like to connect with us, please feel free to reach out at info@nanoimagingservices.com. So thank you again, and we hope to see you next month. Bye, everyone.
Thank you. Bye.